Thank you, Andrea. Thank you very much, and thanks for making me feel like there's another zoologist in the room. I won't try. Oh, nice. Cheers. So what I thought I would do for this talk is kind of tell you about a project that I've been working on for quite a while. And when Andreas told me about the symposium, I thought I'd sort of keep it super baseline level, not get into any jargony business, and just kind of show you how we've been applying transcriptomics to understand some of these evolutionary phenomena that we work on. So I'm an evolutionary biologist, and like most evolutionary biologists, I'm interested in some aspect of this diagram here. So the biggest conundrum in evolutionary biology is how to explain the vast biodiversity that we see on the planet. And so, this is kind of a representation of the tree of life. There are biologists who work at the macro level, so what explains the diversity of the big clades. And then there's geneticists like me who are interested in what happens along the tips here, right? So we want to understand the microevolutionary processes that happen. And I'm going to use this abstraction to kind of explain the framework that I use to study evolution. So the basic level, you get one species that splits into two. This process is called speciation. And so I spent my career trying to understand what are the things that, first of all, make two species. So what are species? This is the philosophical question that I'm not going to get into. But what we want to understand are the processes that happen along these branches. So some genetic changes are going to accumulate along these branches. Ultimately, these species are distinct in some way. Okay? So we want to know what that way is, and we want to understand the genetic mechanism of that. So there's been sort of lots of debates about how to define species and all that. I'll spare you the details. What I use is a very simple definition of what species are. So species are two groups of individuals or groups of populations that don't interbreed. So we're using this criteria of reproductive isolation, okay, as a way to distinguish species. So reproductive isolation is a fairly straightforward process. You can imagine that if two individuals from a population or two separate populations meet, there are several things that need to happen for them to be the same species, right? So they need to be able to mate and fertilize and then produce viable and fertile offspring. So if there are defects in any of this time scale here, that means that there's some level of reproductive isolation. There's some incompatibility, okay? And you don't have to remember any of these terms. Classification biologists love to divide things into classifications. So there's post-zygotic barriers, those that evolve after fertilization, and those that evolve before fertilization. So I've been fascinated by the pre-zygotic aspect of things, and you can imagine those are incompatibilities that involve sort of defects that occur between two genomes, right? So either females won't mate with these males won't mate with these females, there's just, for example, let's take a close to home example. You're not attracted to male chimpanzees if you're female, and female chimpanzees are not attracted to male humans, right? At least 99% of the time, okay? So that would be considered a reproductive barrier. But this can also happen at the fertilization level, right? So these are biochemical incompatibilities that prevent, for example, sperm from one species to interact with eggs from another species. So I've been interested in understanding how these processes work, and I don't want to get too into the theory behind this, but evolutionary biologists are really interested in these kinds of incompatibilities because they involve this evolutionary force called sexual selection. Sexual selection is sort of like natural selection, which I'm assuming people have a decent idea about, but it involves differential fitness in terms of mating or fertilization. Okay, so when you look in the animal kingdom, especially let's say these birds, you notice that some of them have pretty irrational and exaggerated traits. And the thing to notice here is that these are all males that have evolved really dramatic plumage, really dramatic features. And the reason for that is this process called sexual selection. Right? So these traits have no survival value, they only have mating value. And so you can imagine this is a really potent evolutionary force that drives changes between species pretty rapidly. So I've been interested in studying this phenomenon for a while. I study it at the behavioral level, but more recently we've been looking at this at the gamete level. Okay, so this concept of sexual selection at the post-copulatory stage, I'm sorry for the jargon, but all this means is that in cases where gametes can compete for fertilization, you get the exact same sexual selection dynamic. Okay, so it's an analogy of plumage and behavior and things like that, except now we're talking about biochemical processes that are happening between gametes. Okay, so I'm going to tell you how we study this process in some species of Drosophila, and I'm going to give you an example of how we apply transcriptome sequencing to study this phenomenon. Okay, so I work in a group of Drosophila, and I thought Andreas was going to say I work on melanogaster, but he kind of saved it. So I don't work on melanogaster. I actually work on a species down here. We work on several of these species, but today I'm going to tell you about this species of Drosophila, and there's a reason for that. They're very well characterized. We've done lots of studies to understand their biogeography, their evolutionary history. We have genomes. We got everything, right? And so they're an ideal system primarily for this reason. So I showed you this plot describing the types of reproductive barriers that species can evolve. Okay? And on the left here is a table of all the possible pairwise crosses that you can make between those 4 individuals of the species. And so now here what I'm showing you is the strength of isolation at each of these different categorized barriers. And you should notice right away that these gametic barriers tend to evolve very fast and very frequently between those species. Okay? So this species group is very prone to developing these types of gametic incompatibilities. And of course, as a geneticist, I'm interested to know why. Why are they prone to developing these kinds of incompatibilities? Okay, so what is gametic isolation? Okay. Before I go into that, let me just quickly familiarize you with the reproductive system in flies in general, and particularly in these groups of flies. So here's a male reproductive system. I'll color this later. There are 3 main organs you can imagine. So sperm is produced in testes. So this is not unusual. I'm just sort of familiarizing you with the structure of stuff here. Okay. So sperm is produced in testes, and then this organ, known as the accessory gland, is very analogous to the human prostate, produces many, many proteins that are secreted in the male ejaculate during mating. And these contain many, many proteins that we're very interested in, and they actually produce many functions post-mating that we're quite interested in. Okay? And then the female reproductive tract, and this is where the business end of things is, right? There's the gonadal tissue, which produces ovaries, and then there are 2 compartment tissue that stores sperm for very long periods of time. Okay? So this is really important. The reason that they store sperm is that females can actually mate with multiple males over her lifetime and can store sperm from multiple males at a given time. Okay? So what that means is that there's lots of opportunity for competition between sperm from different males to compete for fertilization. And that sort of goes back to where the sexual selection is happening. Okay? So what is the gametic isolation that we see between species? So here's just an example of 2 of those species that if you cross them, so within each species, so here's the names don't matter here. The green species, between them, fertilization rate is pretty high, as you'd expect. I mean, they're the same species, right? Same with this orange species, their fertilization is really high. But now if you cross between species, you see that either you have almost no fertilization or fairly reduced fertilization depending on the direction of the cross. Okay? And we've done a lot of work to show that this is in fact a fertilization defect. This is not an inviability thing. Okay? The details of how it works are completely a mystery to us. We can't tell just by doing normal microscopy, looking at sperm, looking at the fertilization process. We can't really tell what goes wrong. So here's just some of this data to show you that we can tag sperm with GFP and count where they are, and we can kind of compare that to the wild type and the crosses between species. And we really don't see an appreciable difference in the storage and usage dynamic of sperm between or within species. Okay, so the orange bars and the gray bars tend to sort of be the same, and in cases where they're different, it's not an appreciable difference, at least not to the extent that it explains this really dramatic reduction of fertilization. Okay, so when I was a graduate student, I joined a genetics lab, an evolutionary genetics lab, and we wanted to know what are the genes that cause this incompatibility, right? So similar to the talk we heard before, we wanted to map regions of the genome that contain genes that cause these incompatibilities. And so we would do this by just standard QTL mapping. Okay, and here's just, I'm going to summarize to you the things that, so 5 years of work in one slide. They were mostly pleasant years. The way we do QTL mapping in flies, now I showed you that these guys have pretty severe incompatibilities, but we can still get hybrids, and those hybrids are fertile, and we can backcross those hybrids and get so-called backcross individuals that have shuffled genomes, and then we can take these individuals, phenotype them, and ask, are there associations between genotype and phenotype? Okay, so here's just one example of those crosses. We get a clutch of individuals that have mixed chromosomes, and then we ask what is their fertilization success. And similar to the previous talk, we essentially generate QTL plots and find regions of the genome that have QTL. Okay, so here's the QTL that we found on 2 chromosomes. Here's a really big QTL that encompasses the majority of the chromosome. Here's a set of smaller QTL. So it became really clear that there's some genetic complexity to those phenotypes, and here I'm just mapping the male component of things. You can imagine there's a male component, there's a female component. Here we're just talking about the male component. And at this stage, we realized we really couldn't find map regions of the genome using the standard approach. So that's when we sort of started to think about applying proteomics or transcriptomics. I won't talk about the proteomics work here, but we'll just talk about the transcriptomics. So the rationale is very simple. The thing that is incompatible with females that comes from males is either sperm or the seminal fluid that contains the proteins that is transferred. Okay, so it's a very simple logic. Okay, I already told you that sperm is produced in these organs, the testes. Okay, you can see them swimming there. And then the accessory glands is really the business end of things. And we've known from previous work in other species that the accessory glands produce proteins that stimulate a variety of functions post-mating that are important for fertilization success. Okay, those include things like increasing egg production in the females after mating, increasing ovulation, storing sperm, et cetera, et cetera. Okay? So these are sort of our target tissues in many ways. And this final organ produces the mating plug, which is a very ubiquitous feature in many animals. So what we've done is to basically sequence transcriptomes from these 3 tissues and just do the very simple thing of saying, well, if it shows expression bias in that tissue, it probably has some function in that tissue. Okay? And so this is what it would look like. You get, I'm not glossing through the details of how to do this, but if you have, for example, this is a gene that has testis-biased expression, we assume it's a testis-biased gene, and that's going to be under our query, so on and so forth for the rest of them. And then ultimately, we have a set of genes, and you can see there's not many of these accessory gland biased genes. There's tons of testis-biased genes. This is very standard in Drosophila, and then some of the other ones. And so because we're evolutionary biologists, and we know that because these species are so closely related, we're mostly interested in genes that show rapid signatures of evolution. Okay? And without going into the details of how we do that, we essentially look at the rate of amino acid substitution across those different gene classes. And this is not a huge discovery, but the genes that are expressed in the green tissue, the accessory glands, tend to be the most rapidly evolving in the genome. Okay? And here's the genome average in white. And you can see, I'm not showing you any significance here because all of these pairwise comparisons are significant. What we're really interested in, though, is, okay, out of these rapidly evolving genes, where are those on the chromosome and do they coincide with our QTL? So here I've just sprinkled them across the chromosomes. You can see that we have some that are fairly rapidly evolving on that region, and that happens to coincide with one of our QTL. And work I'm not showing you is now we've been targeting the function of these genes, and they do in fact seem to have some effect on fertilization success. Okay? So very simple rationale of just sequencing transcriptomes, we were able to get to several candidate genes, and now we're characterizing those. Okay, so that's the male side, which is arguably the easier, simpler side, and that's what I've done during graduate school and early in my years here. What we really wanted to understand is what happens in the female reproductive tract. And this is arguably the more complicated, more difficult aspect. So let me just tell you the approach that I just described that we use in males doesn't really work here for a couple of reasons. So we do the same thing where we do RNA sequencing of the reproductive tissue and ask what is tissue biased. You get something like this. But female proteins don't show any obvious signature of rapid evolution in our analyses. So here's just a comparison with the genome average. Here are the female reproductive tract genes, and then here are the male reproductive tract genes that I just showed you for comparison. And you can see these are not rapidly evolving, and they're fairly reasonably conserved compared to the genome average and the male proteins. Okay, so that approach of picking candidates based on the signature of rapid evolution is not really going to work. Okay. We then turn to some of our previous knowledge on the reproductive tract. So Mariana Wolfner, who's a colleague of ours here at Cornell, is really the world leader in understanding reproduction in flies. And this review kind of summarizes some of the work out of Mariana's lab and some of the complexities of things that happen within the reproductive tract after mating. And don't squint and read any of it, I'll just point out that there's tons of stuff that has to happen correctly for fertilization to proceed in the normal way. So, things like increasing oogenesis, sperm viability, et cetera, et cetera. So the complexity of processes that happens in the reproductive tract could go wrong in between species matings, right? So what we wanted to do, and by the way, you can kind of have a profile of these just by gene expression. So after mating, females will undergo stereotypical gene expression changes that are ultimately related to these. We don't understand how, but they're ultimately related to these. So we reasoned that we could probe these gene expression changes after a mating with a conspecific male, so males from the same species, or matings with a male from a different species, so heterospecific males. And if there are differences between those regulatory changes after mating, that might give us a clue as to what is going wrong after mating. So we designed a very simple experiment where we did RNA sequencing on females, so females that are unmated, females that are mated to their conspecific males, and females that are mated to the heterospecific males. And the idea here is we really want to know what is different between these two treatments, and obviously this would be our normal treatment. So there are tons of genes that change, about a couple hundred after mating. Here's our unmated sample. This is just a multidimensionality scaling plot where I'm just showing you the grouping of samples. And we did our sampling at 3 different time points, so 3 hours, 6 hours, or 12 hours after mating. And the thing to notice here is that the conspecific and the heterospecific are completely distinct at earlier time points. Okay? And as you go later in time, they become more similar. So conclusion here is that there's a major difference in the regulatory response after mating between the conspecific and the heterospecific cross. Okay? To distill this further, okay, I'm going to now create clusters of those genes that change after mating and group them by expression profile. Okay, so that's what this looks like. So these clusters are based on what the profile of change of that particular gene is across time. And the thing to note here, there's lots of congruent changes between the two mating types, but there are these 2 clusters that are enriched for genes that show a very distinct expression profile. So we wanted to know what those are. So we grab those genes that show this distinct expression profile, and we do Go analyses, which I'm not a big fan of. In this case, it really worked. And it shows us that many of those genes are actually stress response genes or immune response genes. Okay. So what that means is that females seem to be responding to foreign males as if they were a bacterial infection or something. Okay. So what does that mean? So we know a lot about the immune response pathway in flies, and let me just give you a quick vignette about that. So this is well worked out. They even handed a Nobel Prize for it and everything. The way it works is there are receptors in certain tissue in flies that are able to recognize proteins or particles on the bacterial surfaces. These are peptidoglycan receptors. And stuff happens, and at the end, there's this master regulator that upregulates the immune response. Okay? So this regulator, relish, in our data set, is highly responsive to the heterospecific mating, but not the conspecific mating. So that indicates to us that it is initiating an immune response downstream in these heterospecifically mated females. And what they do is they actually upregulate these antimicrobial peptides. So if you look at what these antimicrobial peptides are doing, they are indeed being upregulated. Here's just a sample of those across time, specifically in the heterospecific mating. So these females are really responding to these males as if they were a bacterial infection. Not only that, there are other stress response pathways that get upregulated only in that cross between species. And now this is our sort of hypothesis, the mechanism of how these sperm are essentially being treated as if they were alien objects. Okay? This is not just in the female reproductive tract, but in other tissues, for example, in the head and the ovaries. We can see that this is a complicated plot. But just look at the blue here. These are also immune response or stress response genes. Okay, so what this has done is really reignited an idea that has been in the field, and that is that male proteins can be immunogenic in female reproduction. This idea sort of died a few years back, so these guys actually don't know where they are. They may be retired or something. And that idea now is reignited with our work and some others in the field, and we're now following up on how these immunogenic responses are affecting fertilization success, right? So the idea really from this talk is that we had no concept about the genetic mechanisms, but just by doing RNA sequencing in these different samples, we're able to now generate hypotheses that we can test in the lab. And my lab currently is testing some of these, what these genes and what the consequence of this immune response really is. Okay, I'll spare you the conclusions, since you guys, so more of a plant crowd. And yeah, let me just thank my lab that I used to be in. I already miss it. These guys here, and now I'm at Syracuse. If you have wonderful techs, undergraduates who are looking for jobs, please send them my way. I'm looking to hire a few. Thank you. Yeah.
That was fabulous. I mean, I did my PhD with yourself too, so I
Nice, thanks. I'm sorry you had to go away.
Yeah. So maybe, I mean, you presented, I thought that was fascinating, but you're presenting it as, interesting that the female is responding to these alien male cells as an alien thing, but is it maybe, could you look at the other way, that it's, how do females receive con-specific sperm as, suppressing that immune response? I mean, is there anything like
Yeah, that's a great idea. Yeah, I was actually at a meeting in Sweden last week, and somebody said the exact same thing. Is that it's the conspecific males are adapted to suppressing the immune response in females, and that these foreign males are naive in that mechanism, right? They don't know how to do that. That's still compatible with this coevolution idea between males and females within species, right? And so, because they're failing to suppress this immune response, their sperm is being targeted. So why are they failing? There's some molecules in their seminal fluid that is unable to do that. And we're curious what those are. But yeah, I think the idea is not incompatible with the alternative, which is females are recognizing foreign objects. Yes?
Fascinating talk. So maybe obvious question I'm working on, like, is it also the same mechanism, species frontiers between incompatibility, crossing pigs and cows and stuff like that? Do you think about these mechanisms are conserved and balanced?
Yeah, so there are some cases in mouse, in musculus, for example, where there are gametic incompatibilities. I think the exact type of incompatibility is going to vary in space and time, right? So, and it really depends on the species, depends on the selective pressures. There, in Drosophila, you can find the entire gamut of reproductive barriers, right? These species are unique in that, well, they're somewhat unique in the ones that we know about that have an excess of these gametic incompatibilities. I don't know if you remember that bubble plot. There are some that have some crosses where you find hybrid male sterility, right? There are some where there are female choice at the behavioral level. So it really depends on the history. We've picked on these species just because they probably have a higher rate of what I'm calling this post-copulatory sexual selection, right? So why do they have that? It's probably something to do with their life history strategies, but they're ideal for this kind of barrier. But it'll vary depending on it's so species dependent. So we were not going to come up with rules about speciation just from these guys. Yeah.
You might have already answered this, but basically these species are promoting inbreeding. When you get some exceptional progeny that are the hybrids, genes. Are those hybrids showing better fitness, or I mean, what's the fitness rationale behind it?
So in the particular species pair that I talked about, there is heterosis in the hybrids. So the hybrids outperform the parents in all the sort of viability metrics that we've looked at. They do suffer in some, depending on the cross, in some sperm traits. Okay? But they're bigger, they live longer, they're fatter, right? They're better in some ways, but that's the heterosis component. Now you can use a totally different species pair, and then the hybrids are totally useless. Right? It seems there's no, because they're evolving independently. They're not fine tuning. They're not trying to avoid inbreeding because they don't see each other in the wild, right? Does that make sense?
They're not trying to avoid hybridization.
Yeah, because these are geographically isolated. The ones I talked about.
And are all these Drosophila species, are they all like secrets known of all of these, or partial?
No, but it's 2019, and if it's not known, we just sequence it. That's what we've been doing. So we actually have a massive project with Andy where we're generating a very large coverage PacBio genomes for any species that anybody works on. Really. So if you have a Drosophila, I don't know why you would, but if you have a species that you want to sequence, just let us know or throw it in the machine. You had a question?
Yeah, I'm curious. Certainly, we know in the plant community that recognizing flagella is a really important thing for kind of spotting who the bad guys are.
Yeah.
Have you guys ever looked to see, like, are there differences between the flagella and bees?
In sperm or split?
Yeah, the sperm for the different types of flies.
Yeah, we have. So we've done proteomics on isolated sperm from 2 of the species. They have the same content. There are some sequence differences, but I don't think we can relate them yet to. It's a really good point. I mean, the sperm components of the sperm tail seem to be very conserved across Drosophila. But we haven't looked at functional differences in like amino acid substitution. I'll go look. It's a good idea, but yeah, so far it's not obvious at least. Yeah.
So I don't want to argue the species concept with you, but the biological species concept never worked for plants, right?
Yeah, that's why it's awkward to be here. Yeah, go on.
So I mean, your species like diverged only yesterday.
They did,
and they
yeah.
have this amazing reproductive isolations.
Yeah.
But in plants, they can diverge millions of years and still they can cross. So why do you think it's a difference here?
Okay, I've done that. Plants are, I think species concepts should be applied where plausible. In our case, it's a convenience, right? I mean, you can't argue that the biological species concept is invalid, right? If you can't cross 2 plants, there are 2 species, right? But now then, if you can cross them, but there's a continuum between, that's where I don't have a good answer. But species concepts are really organism specific in many ways. I mean, it works. In our case, like plant, we always are jealous of plants because they stay where you left them. So like if there's, okay, well then maybe they don't. but it's convenient for us. We take examples where there's very clear reproductive barrier, and then it's just a way to study the mechanism of isolation. It's not so much. I mean, I don't really care about species concepts as much as I care about the mechanism. So, for example, also in bacteria, like species concepts are hard to apply in microbes, right? So, yeah, I'm not going to defend the biological species concept, but yeah, use whatever you want and just leave me out of it. Okay. Thank you
So much.